Okay, so then we'll just start by looking at the diagrams here. So we got this first one, which is the digestive system. So we're labeling all these different parts here. So if we start with A, what is A pointing to?
Is it tongue?
I have oral cavity.
Yeah, so A is pointing to the tongue. So if you said tongue or if you said mouth, I'll take either of those for A.
Okay.
All right, what about B?
Liver.
What is B?
I said right lobe.
B is the liver. So what does the liver do?
Well, the
It's
liver.
bile.
Yeah.
Bile?
Right.
Produce bile?
Exactly. So the liver is going to be responsible for producing bile. Perfect. Cool. What about C?
Gallbladder.
C is gallbladder. So what's the gallbladder job?
Storage the bile.
Right. So it's going to be storing the bile. So the liver is going to be producing it, and then the gallbladder will be storing bile. Perfect. What's D?
I have the ascending colon. of the
Ascending
large
colon.
Of
intestine.
large
Yeah. So if you said
intestine.
ascending colon, what the ascending colon is part of what overall structure?
Oh, the
Large intestine?
The large intestine, right. So if you said ascending colon or if you said large intestine, I'll take either one of those. Okay. So what's the large intestine's role in the digestive system?
Doesn't he absorb most of the water?
Right. It's going to be involved with absorbing water, and that's going to help to form your feces. So it's going to be involved with absorbing water mostly, and then it's also going to have kind of a minor role in absorbing different vitamins and things like that too. Yeah.
You got me on that one. Really good on the homework.
Oh, yeah, sorry.
I was like, absorbing the water. I was like kidneys, ADH, and it was large intestine.
Yes. Yeah. Cool. And then with E, what is E pointing to?
Rectum.
Yes, E is pointing to the rectum. Okay. So what's the rectum's job?
It'll take it after it becomes bolus it becomes feces at that point, right?
Right. Yeah. So the feces is going to pretty much accumulate inside the rectum, and then that's going to be the first place where we're going to see the defecation process begin to occur. All right, good. That's E. Then we have F. So what are we pointing to with F?
So that was hard for me.
Salivary glands.
Mm-hmm.
Yes, F was the salivary glands.
Yeah.
Okay. So what do the salivary glands do?
They have a role in the chemical process because they secrete different types of chemicals. I don't remember exactly which chemicals, but I believe there's 3 of them, right? There's
Amyloid.
like hydro.
So there's 3 different pairs of salivary glands, but it's mostly going to be producing what? Can anyone help her out?
Salivary amyloids.
Yes. So it's going to be producing saliva, and inside that saliva, there's going to be an enzyme called salivary amylase. Okay. And so what does salivary amylase break down?
Carbohydrates.
You don't remember? Yes, exactly. So salivary amylase helps to break down carbohydrates. Good. All right, then we get to G. Okay, so what is G pointing to here?
The
The pharynx?
The pharynx.
Yes.
Right. So G is the pharynx. So that's going to be the place where swallowing is going to first occur. Then we have H. What are we looking at with H?
esophagus.
The esophagus. Okay, so what does the esophagus do?
Well, the food goes down to the esophagus. Oh, it connects. The esophagus is a connecting point between the throat to the stomach.
Exactly. Yeah. So the esophagus, its only job is to just transport the food from the pharynx down into the stomach. Right. Yeah. Then we get to I. Okay, so what are we looking at with I here?
The fundus of the stomach.
Right. So I is pointing to the stomach.
Yeah.
And so what does the stomach do?
It
does a few things.
Well, it was interesting when I started reading about it that it's kind of like a reservoir for the food. It actually, I didn't look at it that way. It actually holds it.
Yeah. So one of its main jobs is going to be to store the food and just kind of hold it until it's ready to pass out of the stomach, right? That's going to be one job. Okay. In addition to that, there's also two enzymes that get produced inside the stomach. Anyone remember what those enzymes are?
Is it HCl?
One of them starts with a P.
Oh, pyloric.
So hydrochloric acid, that's the HCl, that's the stomach acid there. So that's going to be also important. Cool. And then there's an enzyme there that also gets produced.
Is it like pyloric?
No, not.
No.
So this is where to see pepsin. So pepsin is an enzyme produced in the stomach, and it gets activated by the stomach acid. And so it helps to break down proteins. Okay. And then we also have gastric lipase, which helps to break down any fats in the stomach too. So that's the stomach there. Then we have J. What are we looking at with J here?
The pancreas.
J is the pancreas, right? So what's the pancreas's job in the digestive system?
Well, it has alpha and beta cells for glucagon and insulin, right?
Yes, but that's more about with the endocrine aspect of it. But in terms of the exocrine functions, what does it produce that helps with the digestive process?
I'm not too sure. Can anybody help me out?
Anyone up or up?
Say that again.
Does it create bile?
So what does the pancreas do? What does the pancreas produce that helps with the digestive process? And I'll give you a clue. The digestive process that the pancreas helps with occurs in the small intestine.
I think there's glucagon and insulin.
No, not so much. That does get produced, but that's again more endocrine function, not as much with the digestive system.
Okay.
Yeah.
Yes.
Well, I could see the little green thing is going in there. Is that the common bile duct? So somehow is it bile that goes from the liver, the hepatocytes in the liver down to the being storage in the gallbladder, and then it somehow goes to the pancreas?
No, so the pancreas doesn't have any role with bile. Okay,
Yeah.
but instead what the pancreas does is it produces a bunch of other enzymes. So the pancreas will produce pancreatic amylase, it'll produce a bunch of enzymes that break down protein, it'll form pancreatic lipase to break down fats. So it's going to be producing the major enzymes involved with breaking down food inside the small intestine. Okay. So once all those enzymes get produced in the pancreas will pass all those enzymes into the small intestine, and then from there, the small intestine can be the place where all those enzymes are going to be working to digest those different food particles. Does that make sense?
Yeah.
Okay. Cool. Okay, so that's the pancreas's job. Then we get to the small intestine. Okay. Sorry, I just kind of gave that one away. So K is the small intestine. So the small intestine's job is going to be mainly with the majority of the digestion and absorption is going to be taking place there. Okay. And then the last part, which is L, is what?
The anus.
The anus, right. And so that is going to be where the feces will be exiting the body from.
So what did you say E was? I missed it.
E?
Yes.
Oh.
Yes, E was the rectum.
Oh, okay. It took forever to figure it out.
Yeah, so E is the rectum, and then the rectum leads into the anus, and then from there you get outside of the body from there.
Okay.
Okay, cool. Any questions with this diagram here?
No.
No? Okay, cool. All right. So now moving to question 7. So here we're labeling the parts of this system. So here for A, what are we looking at with A?
The hypothalamus.
Right. So A, we're looking at the hypothalamus there. The hypothalamus. Okay. B and C are the same thing. So what are we looking at?
Neurosecretory cells.
That's what I have.
Right. These are the neurosecretory cells. Perfect. Okay. Then we hit D. And so what's D? This region here.
Infundibulum.
Yes. D is the infundibulum. So that's the stalk that connects the hypothalamus to the structures down below. Okay. Then we hit E. What is E here?
The posterior pituitary.
Right. E is the posterior pituitary gland. Cool. And then F is the what?
Anterior pituitary.
Right. F is the anterior pituitary. So how do we know which one is the anterior pituitary and which one's the posterior pituitary?
One is larger than the other.
Mhm.
Right. So the anterior pituitary is larger than the posterior pituitary. So that's one way. Okay.
Yeah.
And then also looking at this diagram, what's another way that we can tell which one's which?
Well, don't the neurosecretory cells, even though in this picture they're coming off of the anterior pituitary gland, but don't they only come off of the posterior pituitary?
Kind of. So I think I know what you're saying. So in the posterior pituitary, the
Yeah.
neurosecretory cells will run directly from the hypothalamus all the way down into the posterior pituitary and then release the hormones from there.
Yeah.
For the anterior pituitary, the neurosecretory cells end inside the infundibulum, and they'll drop off their hormones into the hypophyseal portal system, which we see up here in D, and then send those hormones down to F, or the anterior pituitary. So the neurosecretory cells are different lengths in the anterior and the posterior pituitary. Cool. Okay. Any questions with this one?
No.
Okay, cool. All right, so that's that. Okay, so then we get to question 8. So liver cells called blank produce bile. Which cells are these?
Oh, hepatocytes.
Right, these are the hepatocytes. So hepatocytes are going to be the liver cells that are involved with producing bile. Perfect. Okay, number 9. We have parafollicular cells or C cells of the thyroid gland produce the hormone blank, which helps regulate the level of calcium in the blood. Which hormone?
Calcitonin.
Right, calcitonin. What's going on here? So calcitonin, calcitonin, there we go. So calcitonin is going to be the hormone involved with producing or helping to regulate calcium levels. Number 10. So the blank are mainly involved with the absorption of water. Which part of the digestive system are we looking at here?
I got that wrong.
So I say the absorptive cells of the large intestine.
Yes, that would be good. So I was just looking for large intestine, but if you said absorptive cells, that works too. But mainly the large intestine is going to be involved with absorbing water inside the digestive system.
You have to correct that because it is marking it wrong.
Yeah, so I'll take care of that.
Okay.
Cool. Question 11. So the pancreas releases the hormone blank in order to raise blood glucose levels during periods between meals, the fasting state. Which hormone is the pancreas going to release here?
Glucagon.
Glucagon,
Yes.
Correct. So glucagon is going to get released during periods where you haven't eaten for a while. Okay? And so during those periods, your blood glucose levels are going to drop a little bit. And so in order to keep those glucose levels back appropriate, okay, you're going to release glucagon, and that's going to produce a whole series of events that's going to increase your blood glucose levels back up to the appropriate level. So this is one way to regulate your glucose levels. All right, cool. And then question 12, the blank performs most of the digestion and absorption of the GI tract. Which part here?
Oh, the small intestine.
The small intestine, correct. So the small intestine does most of the digestion and absorption within the GI tract. Okay, any questions on any of these so far?
No.
No? Okay, cool. All right, question 13. So question 13 says, Caitlin has type 1 diabetes, which has destroyed her beta cells in her pancreas. How will this affect glucose metabolism if left untreated? So we got A, Caitlin's blood glucose levels will be high because insulin production is insufficient. B, her blood glucose levels will be low because insulin will be overproduced. C, they'll be normal because she still has insulin receptors. Or D, they'll be high because she'll produce more growth hormone. What do we think here?
A.
Yes, A is correct. So her blood glucose levels are going to be high because she's not going to be producing any insulin. So the beta cells are responsible for producing insulin. Okay, if those beta cells get destroyed, you're not going to produce enough insulin, and so as a result, you won't have enough insulin to help stimulate the removal of glucose from your blood. Okay, and so your glucose levels are going to be high inside the blood. Okay. Cool. Okay, good. Okay, any questions on this one?
No.
This one makes sense.
Yeah, it makes
Okay, cool. Okay, cool. Number 14. So which of the following are not produced by the adrenal cortex? So we have adrenocorticotropic hormone, or ACTH, you have glucocorticoids, aldosterone, or androgens. Which ones are not produced by the adrenal cortex?
A.
A is correct. So the adrenocorticotropic hormone, this is not produced in the adrenal cortex. Where is this hormone produced?
In the hypothalamus.
So it gets not produced in the hypothalamus, but you're close. Where does ACTH get produced?
I thought it was the hypothalamus and goes down through the anterior pituitary gland.
So the anterior pituitary is where it gets released from. Remember when we're looking at the anterior pituitary hormones, we have that acronym.
Oh yeah, flat pig.
Flat pig.
Yeah.
Okay. And so the A is where we have ACTH. So these get produced from the anterior pituitary gland. Now, actually, I'll get there. I'm sorry, I'm getting ahead of myself. But actually, while we're here, what are the other hormones that get released from the anterior pituitary?
We have, and so it's going to be F is follicle stimulating hormone.
Right. So with follicle stimulating hormones, that's our FSH.
Yeah.
Okay. Then what about the L here?
LH, the luteinizing hormone.
Right. LH is our luteinizing hormone. Okay. Then we have ACTH.
Yeah.
What's our T?
T. Oh, is it?
TSH.
TSH, right? Thyroid stimulating hormone. Cool. And we have our P, which is?
The P. Oh, is it? No. Any help out there for the P?
Can anyone think of it?
PRL, I think.
PRL, which is, anyone remember what that is?
Is it prolactin?
Prolactin, yeah.
That's prolactin, correct. Yeah, that's prolactin. Good.
And growth
And then the
hormone
last one, our G,
is
growth hormone, RGH.
That's the one, yeah.
Mhm.
Perfect. Cool. Okay, good. So remember, make sure you guys are comfortable with the anterior pituitary hormones and the posterior pituitary hormones. Cool. Okay, good. So that's that. All right, question 15. Which of the following enzymes is produced by the pancreas and digests protein? Okay, so we have trypsin, salivary amylase, pancreatic amylase, or pancreatic lipase.
Trypsin.
Trypsin, yes. Trypsin is one of those proteins that helps to break down, or sorry, one of those enzymes that helps to break down protein. Okay, good. What do salivary amylase and pancreatic amylase break down?
Salivary amylase, isn't it carbohydrates?
Mm-hmm. So salivary amylase breaks down carbohydrates. What about pancreatic amylase?
I'm not too sure.
Don't overthink this one. Don't overthink it.
Okay.
Think of what salivary amylase does.
Carbohydrates too?
Carbohydrates as well. Right. Same enzyme, just in a different place, or a more effective enzyme somewhere else. Right. So pancreatic amylase and salivary amylase both break down proteins, or sorry, carbohydrates. Okay. And then pancreatic lipase. What does pancreatic lipase break down?
Break down lipids, something.
Yes, it breaks down lipids.
Yeah.
Yes. So that one's helpful because when we think about lipases, they will always break down lipids. Okay. That's one way you can always remember that.
Oh, okay.
I just guessed it. Like, oh, lipids.
Yeah, well, there you go. Good. All right, so that's that. Question 16. Okay, so question 16. So when there is no chyme in the small intestine, what happens? So we have A, the sphincter of Oddi closes and forces bile up the cystic duct and into the gallbladder. B, the sphincter of Oddi closes and forces bile up the common hepatic duct and into the liver. C, the sphincter of Oddi opens and allows bile into the duodenum. Or D, the sphincter of Oddi opens and allows chyme to flow into the common bile duct and into the liver.
It's A.
Yes, A is correct. So if there's no chyme in the small intestine, the sphincter of Oddi is going to be closed. And when it's closed, that's going to cause the bile to back up through the common bile duct and then into the cystic duct, and then eventually store that bile inside the gallbladder. Perfect. Okay. Question 17. So antacid tablets like Tums or Rolaids are used to help neutralize the acidity of gastric contents. What cells are responsible for this acidity?
The parietal.
Is it the parietal cells, chief cells, rugae, or mucous neck cells?
Parietal cells.
Right, the parietal cells. So the parietal cells, they're responsible for producing the stomach acid. Cool. Yeah. Okay. Any questions on any of these multiple choice questions so far?
No.
Okay, cool. Okay. Question 18. So describe the process of defecation. Okay. So in order for us to poop, what's the first thing that has to happen?
We have to put food in our mouth.
We'll assume we're in the large intestine already.
Oh, in the large intestine.
Yes.
Well, aren't there like fluids that get released to start the chemical process part?
Not really yet. No, not here. So we'll just kind of begin with these stretch receptors. So inside the rectum, right?
Oh.
Inside the rectum, we're going to have these stretch receptors. And what's going to happen to these stretch receptors?
Oh, the stretch receptors. They cause an effect, don't they? Once they stretch.
Yes, they do.
Yeah.
Yes. So once the rectum begins to fill with feces, I'm going to kind of just start here. The rectum fills with feces, which causes the stretch receptors inside the rectum to begin to fire, right? They start producing action potentials. So when they start producing those action potentials, where are those action potentials going to go?
Well, you were breaking up. Where are those action potentials going to what?
Where are they going to go?
We're gonna go
Where are we delivering those action potentials to?
from the descending colon to the sigmoid colon, and then down the process. So like the rectum and the anus, which causes the rectal muscles to contract, and it creates pressure to open the internal anal sphincter. And then if it's the the external anal sphincter isn't clenched, then the feces exits the anus.
Exactly. The only one thing you missed, and we skipped a little bit of a step here. So these action potentials, they're going to go to the spinal cord, to the defecation center, spinal cord's defecation center. And so the spinal cord's defecation center is going to produce a bunch of action potentials using the sympathetic or parasympathetic division.
Wouldn't it be parasympathetic?
Right. Okay, perfect. Parasympathetic division. And they go down to the rectum, the colon, to the rectum and to the colon, essentially, muscles. Okay. And then from there, like you said, what's going to happen is those muscles are going to begin to contract. Okay. So the rectal muscles and the colon muscles begin to contract. And as they contract, that causes an increase in pressure. And ultimately what happens is the internal anal sphincter is going to relax. Okay. Let me kind of set, so there's another step. So those motor impulses will also cause the internal anal sphincter to relax. And so once the internal anal sphincter relaxes, now we're kind of, you know, in the defecation time period here. In order to allow defecation to fully happen, we need now to relax the external anal sphincters. Okay. And so that one we can control voluntarily. Okay. So when we allow the external anal sphincters to relax, that is going to allow defecation to occur. And that's when I was. So from beginning to end here, yeah. Go ahead.
I misunderstood this question. I thought you were talking about like all the steps to digest the food. So yeah, I misunderstood. So I'm glad we're going over this.
Yeah.
Thank you, Katie.
Yeah. So this will just involve with defecation. Okay. So in order for defecation to occur, you first got to have the feces move from the sigmoid colon into the rectum. Okay. Once the rectum begins to stretch, it's going to activate some stretch receptors. Those are going to send impulses down to the defecation center inside the spinal cord. And then from there, we're going to have some motor impulses along the parasympathetic division going to the rectum and the colon muscles. Okay. That's going to cause the rectum and colon to contract and increase the pressure. And then we're also going to have some motor impulses going to the internal anal sphincter. Okay. And it's going to cause that sphincter to relax. And then the last thing that's going to happen is the external anal sphincter is going to relax when we're ready to actually poop. And then that's going to allow defecation to take place. Cool. All right, how do we feel with this whole process here?
I'm going to have to revisit that.
I think it's not.
Cool.
It's okay. It's not so bad.
Yeah. So when you kind of look at it and you kind of if you draw it out a little bit too, it helps to kind of make a little bit more sense and it becomes a bit easier to understand.
Okay.
Okay. So that's that one. Okay. So then we have question 18. So question 18, we want to look at how the hormones from the anterior pituitary and from the posterior pituitary are going to be released. Okay. So let's start with the anterior pituitary. So if we want to release hormones from the anterior pituitary, what needs to happen? What's the first thing that we need to do?
Well, the hypothalamus, isn't there inhibiting and excitatory hormones, and the hypothalamus will communicate to the anterior pituitary when it's time to either release inhibiting or the excitatory hormones?
Yes. So the hypothalamus is going to have these neurosecretory cells that are going to be able to produce either releasing hormones or inhibiting hormones. In this case, because we want to release some hormones, we're going to be looking at releasing hormones. Okay. So they're going to secrete releasing hormones. So when we secrete those releasing hormones, where are they going to be dropped off into?
Oh, the hypophyseal. We saw it in the.
Yes, they're going to be dropped off. Oh yeah, so into the hypophyseal portal system, which is that network of capillaries inside the infundibulum. Okay. So once those releasing hormones go from the hypophyseal portal or enter the hypophyseal portal system, where do they go now?
They travel to the anterior pituitary.
Right, they travel to the anterior pituitary. Travel to the, I'll just put AP for anterior pituitary. So they travel to the anterior pituitary, and then they bind onto their receptors. Okay. Once they bind onto the receptors, then they're going to trigger the anterior pituitary to release its hormones. One thing I forgot to mention earlier here at the top is in order for these neurosecretory cells to release their releasing hormones, what needs to happen inside those neurosecretory cells? What do we need in order to start exocytosis of these hormones here?
The vesicle comes, right? There's a vesicle involved.
Right. So in order to do this whole process, we first need an action potential, right? And so once we have that action potential, that'll ultimately lead to exocytosis of those vesicles. And then we'll be able to secrete those hormones into the hypophyseal portal system. Okay, so that's the anterior pituitary. What about for the posterior pituitary?
That one's a little bit.
How is this process going to be a little different?
Well, because the posterior pituitary, it doesn't release any hormones. It's merely just used for storage for the hypothalamus.
Close. So the hormones do get released from there, but the posterior pituitary doesn't produce any hormones, right? So what's going to happen here is we're going to have some hypothalamic neurosecretory cells produce those hormones either. Actually, let me ask you guys, what hormones get produced or get released from the posterior pituitary that are going to be produced here in the hypothalamus?
Oxytocin and ADH.
Right, oxytocin and ADH. Okay, so those neurosecretory cells are going to produce oxytocin and ADH. Okay, again, we're going to have an action potential. Okay, and so that action potential is going to then deliver, or is going to cause the exocytosis again. And then from there, we'll be able to deliver those hormones into the general circulation out of the posterior pituitary. Exocytosis from the I'll just put PP for posterior pituitary. And into the general circulation.
So George,
Yeah.
the posterior pituitary gland does not produce oxytocin ADH, right? It doesn't produce it.
Right. Right. So the posterior pituitary does not produce any hormones. Okay. All those hormones are getting produced inside the hypothalamus. But again, remember those neurosecretory cells are going to run from the hypothalamus directly into the posterior pituitary. And so when it's ready to have an action potential, okay, it's just going to release those hormones from the posterior pituitary and into the general circulation. Okay. So the posterior pituitary's only job is just to store and release those hormones, but it's not going to be producing them. They're going to be produced inside the hypothalamus and then transported to the posterior pituitary.
Okay, I made a
Does that make
mistake.
sense?
Yeah, I just made a mistake because I said it was produced in the posterior, but okay, for sure. Okay, now I understand a little bit.
This was tough.
Yeah. So it's a lot of things at work here. So this would be another one another good one to draw out if you're having difficulty with this.
Yeah, definitely.
Yeah. Okay. Last one is to describe the route traveled by bile from the time it is secreted until it enters the duodenum. Okay. So we'll start with the liver. So when the liver produces the bile, okay, where is it going to go first?
It's collected by the bile canaliculi.
So the bile canaliculi, right? Canaliculi. Okay. And then from the bile canaliculi, they're then going to move into the what?
The ductiles. The bile ductiles.
Right. The bile duct. Okay. And then, or bile ductules. Yeah. Okay. And then from there?
The bile ducts.
The bile ducts, perfect. And from the bile ducts, where do they go now?
The right and left hepatic ducts.
Right. Cool. So the right and left hepatic ducts. Okay. And then from there, where is it going now?
The common hepatic duct.
All right. So they'll join together to form the common hepatic duct. Cool. And then from there, the common hepatic duct will do what?
From the cystic duct.
Right, so it'll join the cystic duct and then form what?
The common bile duct.
The common bile duct. And from the common bile duct, it then has to pass through what opening before it can enter the duodenum?
The sphincter of Oddi, right?
Yes, the sphincter of Oddi. Sphincter of Oddi, and that will bring the bile directly into the duodenum.
Duodenum.
I
Okay.
misunderstood.
Any questions with anything we've looked at so far? I know this was a lot. There was a lot of material in this last week here. Any questions?
Yeah. Damn. I made a mistake on the homework then. I misunderstood it. I misunderstood this question.
That's okay. That's okay. It's better to make the mistake on the homework than to do it on the exam. So
For sure, yeah.
Yeah, it's not bad. Don't worry about it.
For sure.
Okay. All right. So if there's no questions, we'll go ahead and jump into today's material. So today we're going to be going over actually before we start, sorry, one thing. So this is our last week before we get into finals week. So we still have 2 more chapters we're going to be going over. We're going to be hitting the urinary system and then the reproductive system, which are chapter 25 and 26. Is everyone able to see Canvas again?
Yeah.
I think so, yeah.
Yes.
Okay, so because we just completed our last homework assignment for the semester, I didn't want to leave you kind of high and dry as far as, what to study for these last 2 chapters. So if you go to the exam study guide section, at the bottom, there's some practice assignments for chapter 25 and 26. There's kind of one that you can kind of do on your own, and then there's a key over here as well. And so this will kind of get you kind of familiar with what you should expect to see regarding those chapters on the final. Okay. So when you're studying for those, so this is not going to be due, it's not anything that's mandatory, but this is just something that you can use to kind of help yourself with studying for chapter 25 and 26. You can use this to practice, and again, that should give you an indication of what to really be focusing on for those chapters for the final. Cool.
Sure, cool. Thank you.
That's good. Thank
No
you.
problem. No problem. Okay, I think that was the big thing I wanted to mention. Okay, then, so today we're going to be hitting the urinary system. Oh, sorry, one last thing before I forget. So if you haven't already, so my mind's all over the place. I apologize. Couple quick things here. So number one, if you have not completed your career essay, that is going to be due today. So that is due tonight. So if you haven't done that, make sure you submit that later today. Again, you can submit that on Canvas, or you can just email that directly to me. Either one is good. Okay. So that's due today. We will still have labs that will be due this week. So we'll have labs due this week for these 2 chapters. And then next week, we will have our review session. So again, that's not going to be mandatory. But if you need help, if there's anything that you're not comfortable with for the exam, okay, the review session will be Monday at 5 o'clock, same time as our normal class period. And so we'll just kind of go for maybe an hour or 2, try to get all of our questions kind of squared away. If you have any questions from the chapter 25 and 26 practice, we can answer those as well. So that's that. And then the final exam will be on Wednesday. And so that one, we'll open that one up at 8 a.m. and then you have until 11:59 to complete it. Cool. So any questions on anything that's coming up?
No.
No.
Okay. All right, so again, if you have not done the career essay, make sure you do that because that is worth 50 points. And if you turned in late, I'm going to have to take off, or sorry, I'm going to have to dock points, and I don't want to do that. So career essay, that's going to be due today. So Erica's question is, do we log in on the last day? Or do we log in? Yeah, do we log in on the last day? So if you're talking about on Wednesday, yes, you can still log in on Wednesday, and you can do that. As far as next week for finals, you don't need to log into Zoom. We're not going to be doing anything on Zoom other than the review session. So the review session we'll do on Zoom just like we normally do for the exam, for the final exam. You don't need to log in. Just go onto Canvas, take the exam. Once you're done, and that's it. Cool. All right. Any other questions on what's coming up?
Lab 21, the urinary system, that's going to be due today before midnight.
That's going to be unofficially due at midnight, but again, if you can just get it done by the end of the week, that's fine too. Yeah. But the big thing is the career essay is going to be due today. Cool. All right. Any other questions?
No.
Okay, cool. All righty. So again, I'll kind of give a few reminders, as far as the review session and the final over the next week or so. Okay, so that's what's coming up. Okay, so now we're going to be doing with the we're going to be learning about the urinary system. So this is chapter 25. Before we start, what's the main job of the urinary system? What do we think?
It gets rid of waste.
Gets rid of waste, right? And so as we get rid of waste, we're going to be producing urine, right? We're going to be producing RP. Okay, so that is actually more of a kind of a byproduct function of the kidney's major overall function. Okay? So one thing that people don't normally think about is the main job of the kidney is to regulate your blood's homeostasis. Okay? It wants to regulate all the levels inside your blood. Okay? And as it regulates all the composition, your blood composition, it's going to be able to get rid of anything else that is not needed. Okay, any excess stuff that does not need to be in the blood is going to end up kind of becoming your waste or becoming your urine. Okay? And so that's going to be how we see the last function there. So we'll piece apart how that is actually going to work out today. So this will actually be kind of another kind of interesting one too as well. Okay, that's what's coming up. So that's going to be what the urinary system's job is going to be doing. So what we're going to be doing today is we're going to be taking a look at the major functions of the urinary system. We're going to be looking at the kidneys and how the kidneys is going to be responsible for regulating your blood composition and how it's going to be producing urine in the process. And then we're going to be taking a look at all the different functions that can take place inside the kidney, specifically inside the nephrons. Okay. We're going to be looking at filtration, reabsorption, secretion, and then we're going to look at the rest of the urinary system and look at kind of the functions that are going to be forming. And then at the last component here, we're going to be looking at how we're going to be able to pee, okay, how our urination is going to actually occur. Okay, so that's the game plan. Okay, so first off, starting with the urinary system. The urinary system is going to be made up of two kidneys. So we're going to have two kidneys on either side of the body. Okay. And then coming out of each kidney is going to be a long tube called a ureter. And so these ureters are going to be leading from the kidneys down into a single urinary bladder. Okay. And then at the end of that bladder is the urethra, which is going to allow the urine to come out of the body. Okay. So throughout this whole process, we're going to be forming our urine, but we're also going to be regulating our blood composition. Okay. So the study of how the urinary system works, as well as the male reproductive system, and we'll look at this on Wednesday, that is called the study of urology, or the medical branch of urology. Okay. So what the kidneys are going to be doing is they're going to be receiving a bunch of blood. Okay. They're going to be receiving about 25% of your blood volume, okay, at any point in time, and it's going to be filtering through that blood. Okay. Most of the stuff that gets filtered out of the blood is going to get returned back into the blood. Okay. But anything that wasn't returned back to the blood is going to exit the kidneys and will end up making up your urine. So that's going to be made of any water or any excess solutes that got produced, okay, or that got excreted out. Okay. So that is going to form the urine. So this is kind of what our kidneys look like in a cadaver. Okay, so here we've got one kidney over here, another kidney over here. You can see the kind of the renal arteries leading into there. You can see the ureters kind of leading out of the kidneys and so on and so forth. Okay. So that's what it would look like in a cadaver. Okay. So when we look at the overall functions of the kidneys, we're going to see that the majority of these functions have to do with regulating what's going on inside the blood. Okay. And so the first thing that the kidneys are going to be able to do is it's going to be able to regulate your blood ionic composition. Okay. It's going to regulate the ions inside your blood. Okay. So the most important ones that it's going to be able to regulate are going to be sodium, potassium, and then chloride. So what's going to be happening here is, let's say you're at home, you're sitting watching a movie and you're eating just a bunch of pretzels. Okay. Those pretzels are going to be pretty salty, and so your salt concentrations are going to start to increase. Okay. You're going to have lots of sodium inside your blood. Okay. Well, naturally, our blood wants to keep a consistent level of sodium. Okay. But if we're eating a bunch of pretzels, our blood sodium levels are going to increase. So what the kidneys are going to do is they're going to start to filter through the blood. Okay. It's going to retain as much sodium as it wants. Okay. And then whatever else is going to end up getting excreted out as your urine. And so in doing so, that's going to help to bring these sodium levels back down to the appropriate level. Okay. And it's going to do the same thing with potassium and same thing with chloride. Okay. So if your blood compositions get too high, it's going to be able to pee. You're going to be able to pee those ions out and then bring your levels back down to normal. But if they're too low, you're going to be able to retain as much of that stuff as possible in order to keep those levels as close to normal as possible. So that's going to be able to regulate your blood ionic composition. Okay. At the same time, it's also going to be able to regulate your blood pH. Okay. And so in order to do so, it's going to be able to allow you to excrete any extra hydrogen ions if your blood is too acidic. Okay. So if our blood gets too acidic, our blood pH is going to drop, okay, because there's too much acid there. So what we can do is we can excrete some hydrogen ions, excrete some extra acid. And in doing so, that's going to bring our blood pH, again, back up to our normal level. Okay. If our blood pH gets too high, okay, then we can excrete some bicarbonate, which is kind of a base. Okay. And so by excreting out that extra base, okay, again, we can bring our blood pH back to its normal level. So it's going to be multiple different ions that are going to be able to do different things there. Okay. The third thing it's going to be able to do is it's going to help to regulate your blood volume. Okay. So if there's extra water inside your blood, okay, well, then what you can do is you can just retain as much water as you need and then excrete the rest. Okay? And then that's going to help you to kind of maintain your blood volume at an appropriate level. Okay? The other thing that is useful for is it's helpful with regulating your blood pressure. Okay? So if you have extra water inside your blood, okay, you're going to have extra water inside your blood. That's going to cause some extra blood pressure because you're going to have more stuff pressing on the walls of your arteries. Okay? And so in order to kind of combat that, we can excrete some of the extra water through the urine. Okay? That helps us to reduce our blood volume, and as a result, that'll reduce our blood pressure. Okay? And so we can kind of facilitate that there as well. Okay? So that's one way that we can kind of facilitate our water retention or kind of water excretion there. There's also a few enzymes that can get released. One of them is called renin. And so renin is an enzyme that helps to regulate your blood pressure, and that gets released from the kidneys. Regulating your blood osmolarity, that's just the amount of solute that's dissolved inside your blood. Can be able to regulate that through the same process as we saw with the ions, as well as with glucose and the various other particles as well. It's going to be able to produce some hormones like calcitriol and erythropoietin, which we saw earlier in the semester, which helps us to produce extra red blood cells. There is regulation of blood glucose levels, and so the kidneys are actually able to produce extra glucose using glutamine as an amino acid. And then the last thing is it's going to be able to produce or excrete lots of waste. Okay? Anything that's not useful for the body or any foreign substances are going to end up getting excreted as the urine. Okay? So the functions of the kidneys are pretty vast. There's going to be a lot of different functions that the kidney is going to be able to do. But again, its overall job is to help regulate your blood composition and your blood homeostasis. Okay? As it does that, it's going to be kind of producing urine kind of as a byproduct of that main overall function in terms of producing or regulating your blood homeostasis. Cool. Any questions on these functions here? Okay, cool. Okay, so as we take a look at the kidneys are going to be the major components that are going to be responsible for producing the urine and regulating our blood composition. Okay? So these kidneys are these retroperitoneal organs, which means that they sit at the back of the abdominal cavity, and they're protected by the 11th and 12th ribs. Okay, these are the floating ribs, if you remember from chapter 7, I believe it was. Okay, and so this is kind of a double-edged sword, because even though those ribs kind of protect them, it can also possibly be a little bit dangerous, because if those ribs get broken, they can actually slice open the kidneys and then kind of release lots of blood and release lots of toxins into the abdominal cavity. Okay, so it's kind of a double-edged sword in terms of protection, but also can also cause a little bit of damage here. So the kidneys are about the size of your fist, about 4 to 5 inches long, and about 2 to 3 inches wide. So if you kind of take your fist and put them just kind of right above your, I guess right below where your ribs would be, that's where you would find your kidneys, and that's pretty much the size that they would be. The right kidney tends to hang a little bit lower than the left kidney, and that's just because the liver occupies a large amount of space on the right side of the body. And so the kidney has to hang a little bit lower just because of the placement of the liver. And so when we look at the study of the kidneys and all its anatomy, its physiology, and any diseases associated with the kidney, that's the study of nephrology. So those are the kidneys. Okay. So when we take a look at the external anatomy of the kidneys, there's going to be this little indentation here at the medial aspect of the kidneys. This indentation is called the renal hilum. Okay? And so in this renal hilum, this is going to be a location where we're going to find the renal arteries, the renal vein, we're going to find the ureter, lymphatic vessels, and a bunch of nerves all entering and exiting the kidneys from this region. Okay? So all of those different substances, and the ureters included, are all going to be entering and exiting from the kidneys from the renal hilum here. All right, so that's that. Okay. Then kind of surrounding the kidneys, there's going to be multiple different structures that are going to help to kind of protect it. The first structure that surrounds the kidney, the deepest structure that surrounds the kidney, is called the renal capsule. Okay, and so the renal capsule is a layer of this very strong, very dense connective tissue. And so that forms like the outer coat of the kidney, as well as the outer coat of the ureter. Okay. And so what the renal capsule does is it helps to kind of serve as a protective role in terms of protecting the kidney against any kind of blunt trauma. And it also helps to maintain the shape of the kidney as well. Okay. So that's the renal capsule, and that's the kind of the outer piece of connective tissue around the kidney. Okay. Then surrounding the renal capsule is the adipose capsule. So the adipose capsule is made up of mostly adipose tissue, mostly fat. So again, what that fat does is it helps to kind of protect the kidney, kind of cushion it, and it also kind of holds it inside the abdominal cavity. Okay. And the outermost layer of these kind of protective structures here is called the renal fascia. The renal fascia is another kind of thin layer of connective tissue, and that is going to essentially just anchor the kidney to the abdominal wall and just kind of holds it in place there. Okay. So we got the renal fascia on the outside, the adipose capsule in the middle layer, and then inside we're going to have the renal capsule. And again, that renal capsule is going to surround the kidney itself. All right, so that's that. Anyways, that's the external anatomy of the kidney, and nothing too special there. The internal anatomy of the kidney is where things get, there's kind of a lot going on here. This is where all the action is going to be taking place. Okay. So there's going to be 2 distinct regions. The first one is called the renal cortex. So remember, when we think cortex, we think the outermost, most superficial regions. Okay. So the renal cortex is going to be the most superficial region of the kidney. Okay, this outermost region of the inside of the kidney, I guess here. Okay. So this is our renal cortex. Okay. Then the deeper region is where we find the renal medulla. Okay, so the renal medulla is going to be this entire deep region, or this middle region of the kidney. Okay. Making up the renal medulla are these little triangular structures called renal pyramids. Okay, so this would be one renal pyramid here. This would be another renal pyramid here. Then a third one, a fourth one. Here's another one, and there's another one. So these are all renal pyramids. These triangular kind of structures here. Okay. And at the apex of each renal pyramid is what's called the renal papilla. Okay. And so the renal papilla is going to be the apex that's going to point towards the renal hilum. Okay. So this would be the papilla, this would be another papilla, here's another one, fourth one, fifth one, sixth one. So all the renal papilla are going to be pointing and oriented towards the renal hilum. Okay. And then in between each of the renal pyramids, okay, is a region called the renal column. Okay. The renal column is actually part of the renal cortex that just dives in between these renal pyramids. Okay. And so they form these little columns here that we call renal columns. Okay. All right, so that's that. The next region that we're going to have is called the renal lobes. And so each individual renal lobe is made up of one renal pyramid. Okay. It's made up of the overlying renal cortex, as well as half of the adjacent renal columns. Okay. So here we have a pyramid, here we have the overlying cortex, and then we have half of the renal column on one side, and then half of the renal column on the other side. So all of this would comprise one single renal lobe. So that's the one renal lobe. Okay, then we have the renal parenchyma. Okay, so the parenchyma is just the functional portions of the kidneys. Okay, this is going to be where the filtration and kind of the major actions of the kidneys are going to be taking place. Okay, and that's just the location. Okay, now the specific structures that are going to be involved with the functions of the kidneys are called nephrons. Okay, and so in the renal parenchyma, there's going to be about 1 million nephrons, which are the functional units of the kidneys. These are the actual parts that are actually doing the job here. And we're going to be looking at the nephrons and how they're going to be built and what they're going to be doing later on. Okay, so there's about a million nephrons inside each individual kidney. Okay, and those are the functional units of the kidneys. Okay, and so that's what we see here. So this would be one nephron, okay, that will eventually lead out of the kidney here. Okay, so inside the nephron, the blood is going to be filtered, okay? And so that filtered fluid, which we call the filtrate, is going to move through the nephron, and eventually it's going to exit out of the nephron through the papillary duct. Okay? And so the papillary duct exit out of the nephron through, or kind of exit out of the nephron through the renal papilla, okay, of the renal pyramids here. Okay? So this would be a papillary duct. Okay. From there, the papillary ducts will then lead into a kind of a cup-like depression called the minor calyx. Okay. And so we have multiple of these minor calyces, okay, that will again kind of open up into each individual renal pyramid, or sorry, renal papilla. Okay. Okay. And so that's going to be the next place where that fluid is going to be entering into. Okay. From there, all that filtrate is going to move from the minor calyx into a larger opening called a major calyx. Okay. And so we have a few of these major calyces here. Okay. And at this point, the filtrate now becomes urine, and now becomes considered urine, because no more reabsorption of the filtrate is going to be taking place. Okay. That's going to be at the major calyx. Okay. And then from there, the urine is then going to drain into a very large cavity called the renal pelvis. Okay. Once it enters the renal pelvis, then the urine will then move out through the ureter and then get directed to the bladder from there. Okay. Okay. So all of this is going to be taking place inside the renal hilum. Okay. And so in order to kind of hold all of these individual structures, there's going to be a little bit of a cavity here called the renal sinus. Okay. So the renal sinus is this cavity that contains the renal pelvis, the calyces, as well as all the branches of the renal arteries and the renal veins and the nerves and everything. Okay. Okay, cool. All right. Any questions on the internal anatomy of the kidneys? I know there's kind of a lot going on. Any questions here?
Not right now.
Okay. All right. So now we're going to take a look at the blood supply of the kidney. So as we talked about before, the kidney is going to receive about 25% of your blood flow. Okay. So you have about 5 liters of blood. Okay. About 1.2 liters of that is going to be directed to the kidneys every single minute, okay, in order to get filtered. Okay. Now, in order to get that blood to the kidneys, the blood is going to be moving into the renal artery. Okay. The renal artery directs the blood into the kidneys. And then from there, the renal arteries will then divide into the segmental arteries. These segmental arteries are just arteries that will supply different segments of the kidney with oxygen and nutrients. Okay. Then from the segmental arteries, they're going to branch further into the interlobar arteries. Okay. And so these interlobar arteries are these branches that are going to pass into the renal lobes, or sorry, in between the renal lobes through the renal columns here. Okay. And then from there, these interlobar arteries are going to arch over the renal pyramids, okay. And they're going to form the arcuate arteries. They're going to basically form an arc, okay, over the arcuate arteries. Okay. So that's what we see over here, kind of in purple. Okay. And then from there, the arcuate arteries are going to kind of radiate outwards and produce these extra smaller little branches, and those are going to form the cortical radiate arteries. Okay. Cortical radiate because they're going to radiate into the renal cortex. Okay. And again, they're going to just deliver oxygen and nutrients to those particular areas. Okay. Now, when they enter the cortical radiate arteries will then divide even further into the afferent arterioles. Okay. So we're starting with the renal artery, branch into the segmental arteries. The segmental arteries will branch into the interlobar arteries. The interlobar arteries kind of arch over the renal pyramids and form the arcuate arteries. And the arcuate arteries will then kind of radiate outwards and form the cortical radiate arteries. And then from there, these cortical radiate arteries will form the afferent arterioles. From there, those afferent arterioles will then kind of lead into the nephrons. Okay? So each nephron is going to receive one afferent arteriole, okay, which we see right here. The afferent arteriole is going to kind of branch further and divide into a very extensive capillary network that's kind of shaped like a little, like a ball of yarn, called the glomerulus. Okay, and so this is the glomerulus here. Okay, it's kind of a tangled ball-shaped capillary network. And so what's going to happen now is once the blood leaves the glomerulus, okay, the glomerular capillaries are then going to reunite again to form the efferent arteriole. Okay. The efferent arteriole, and one thing that's kind of interesting and really important about the efferent arteriole is that the efferent arteriole has a smaller diameter than the afferent arterioles. That may seem like a kind of an unimportant detail, but this is actually going to be a really important detail, and we'll see the importance of it later on. But basically what happens here is because the efferent arteriole is much smaller than the afferent arteriole is, it creates a larger blood pressure, a higher blood pressure inside the glomerulus. Okay? And so the glomerulus is going to be the part of the nephron that's responsible for filtration, filtering stuff out of the blood. Okay? With a higher blood pressure inside the glomerulus, you're going to be able to push more stuff out of the glomerulus and then into the nephron here. So it's kind of like if you were to walk into a classroom and you've got, 5 doors as entrances, 5 entrance doors, and then only 1 exit door. Okay, you can have a lot of people entering, okay, but it's going to get real congested because you only have 1 exit door. So it's going to create a lot of pressure inside that room. Okay, same thing is going to be happening inside the glomerulus. Okay, because the efferent arteriole is much smaller, you have a smaller exit point than an entry point. Okay, you're going to end up with a lot of pressure inside the glomerulus, and that's going to cause increased blood pressure and ultimately lead to increased filtration inside the glomerulus. Okay. And again, this will make more sense as we kind of piece everything apart later on in the glomerulus part. Okay, so that's the efferent arteriole. Okay, that's going to be where we're going to be exiting the nephron from. Okay, so you enter at the afferent arteriole, exit at the efferent arteriole. Okay. The efferent arterioles will then kind of divide further into the peritubular capillaries. And so that's what we see down here. And so these peritubular capillaries are going to wrap around the kind of tubular portions of the renal, of the nephron, okay? Specifically inside the renal cortex. Okay? Now, if there are any parts of the nephron that are located inside the renal medulla, okay, then these peritubular capillaries, these efferent arterioles will branch into the vasa recta, and they basically do the same thing as the peritubular capillaries. Okay, nothing special about it. Okay. Okay, so this is one thing I do want you to be comfortable with for the exam. I do want you to be comfortable with the pathway of this blood flow. Okay. So you go from the renal arteries, the segmental arteries, interlobar arteries, arcuate arteries, cortical radiate arteries. Then you hit the afferent arterioles, glomerulus, efferent arterioles, peritubular capillaries. Okay. You can pretty much stop there. That's all I want you to be comfortable or be familiar with. Okay, because as you pass out of that, you just reach the veins, and the veins have the exact same names, just in the reverse order as what we saw previously. So peritubular venules, cortical radiate veins, arcuate veins, interlobar veins, renal veins. Don't worry about these bottom ones, but I do want you to be comfortable with these top ones, from the renal artery down to the peritubular capillaries. Cool. Okay, any questions so far on the blood supply here? No. Okay. So again, this is all going to kind of make more sense. We're just kind of introducing all the players that are going to be responsible here. Okay, so that is that.
So we're gonna go more.
So question. Oh, sorry, go ahead.
Oh, I was saying we're gonna go more in depth then into these.
Yeah. Yeah, so we're going to go more in depth, and this will all kind of make a lot more sense as we kind of illuminate all of their roles here. Yeah.
So,
Okay. So question here. Oh yeah, go ahead.
So at some point, they up to the peritubular capillaries. At some point, they connect to the veins and take the waste out.
Exactly. Yeah. So as the capillaries end up exiting out of the nephrons, they'll eventually start joining together as the peritubular veins, and then those veins will start to converge with one another until ultimately they become the renal vein and then exit out of the kidney from there.
Okay.
Yeah. Any other questions here? Okay. A question here. So in the figure shown, what structure is indicated? Okay. Is this the renal column, the renal medulla, the renal pelvis, renal sinus, or the renal pyramid?
C.
Is it the renal pelvis?
Yes, this is the renal pelvis. Right. So the renal pelvis, that's going to be the location where all the urine is eventually going to converge into and then eventually exit out through the ureters. Okay, next question. So which of the following is the correct order of blood flow? So I'll let you guys take a look at this one and then let me know what you think here.
B.
All right. Did I hear E? Did I hear D?
B. Yeah. Say B. Because you said.
Okay. Anyone else? Elijah says D. Anyone else?
Yeah, we begin with the afferent arterioles of the A, so definitely D is.
Yes. So D is correct. Yeah. So we have the afferent arterioles, then we have the glomerulus, the efferent arterioles, and that'll lead us into the peritubular capillaries. So one way that you can kind of remember which of these two comes first, kind of in terms of the glomerulus, the afferent arteriole starts with A, so A comes before E. So afferent, then you have the glomerulus, and then you exit out through the efferent arteriole. Okay. So A before E, always, in this case here. Okay, good. Okay, now that's going to lead us into our nephron. So remember, the nephron is the functional part of the kidney. This is the location where we're going to be doing all of our filtration and all the kind of action here in terms of either determining what's going to be reabsorbed or what's going to be kind of kept as the urine is going to be taking place. Okay. So the nephron is going to have two different components to it. The first part is called the renal corpuscle. Okay. And so the renal corpuscle is going to be the location where we have the blood filtration occurring. That's going to be this region up here. Okay. And then we're going to have the renal tubules. Okay. So the renal tubules is going to be this later portion where we have the where the filtrate is going to be passing through. Okay. So starting with the renal tubule, or sorry, the renal corpuscle. The renal corpuscle is going to be made up of the glomerulus, which is that capillary network we saw before, okay, as well as Bowman's capsule. Okay. And so Bowman's capsule is kind of a piece of epithelial tissue that's going to surround the glomerular capillaries. So the glomerular capillaries, and then the Bowman's capsule, which surrounds it. Okay, it's kind of almost like a tube that surrounds that capillary network. Okay. Then for the renal tubules, okay, this is going to be the location where the filtrate is going to be passing through. There's going to be kind of three or four major regions. The first one is the proximal convoluted tubule. That's going to be the first region that exits out of the renal corpuscle. Okay. So the proximal convoluted tubule. Okay. Then we have the loop of Henle, or sometimes called the nephron loop, okay, which is right here. And then we have the distal convoluted tubule, okay, which is the end portion here. Okay. And so when we look at the proximal convoluted tubule, the distal convoluted tubule, and the glomerulus, okay, or sorry, the renal corpuscle, these are all going to be found inside the renal cortex. Okay. So not inside the renal medulla, but they'll be found in the renal cortex and the outside portions of the kidney. Okay. After our distal convoluted tubule, okay, we're going to have multiple distal convoluted tubules from multiple different nephrons, all converging into a single collecting duct. Okay. And then from there, we can have multiple collecting ducts, which will then unite together, and they'll eventually form the papillary ducts, which will eventually drain that urine into the minor calyces. So this is kind of a general schematic of what the nephron would look like here. Okay. Let me kind of grab my whiteboard over here, because we're going to kind of revisit this real quick. So this is one thing I do want you to do real quick. So if you're taking notes, go ahead and draw this out. As I draw it here. So just kind of draw out what the nephron looks like. First thing we're going to have is our glomerular capillaries here. Again, we're going to have our efferent arterial, and then we're going to have our Bowman's capsule. We're going to have our nephron loop, or loop of Henle. Oops. All right, so that's that. That's what our, sorry, that's kind of an ugly picture there, but this is everything there. And so if we kind of draw everything out here, this would be Bowman's capsule. This region would be the proximal convoluted tubule, up here. Down here, we have the loop of Henle. I'll just put LOH for loop of Henle. Okay. Then we are going to have the distal convoluted tubule over here. Okay, that's going to be this portion there. And then the last region over here is our collecting duct. I'll just put CD for collecting duct. That's our collecting duct. Okay. One thing to keep in mind here, though, is that even though we have our capillaries, this is our afferent arterial, let's put AA there, and then our efferent arterial over here. Okay. These are eventually going to lead into our peritubular capillaries. Okay. Okay. So I'll just draw this as just a bunch of blood here. Okay, is everyone able to see this picture okay?
I can see the top part.
Yeah, I can see it.
Okay, cool. Okay, so that's what our loop of our nephron is going to be looking like. So let me just kind of draw our right nephron up here. Okay, so these are all the different parts. Be comfortable with these parts, and again, as we kind of go through this, we're going to be kind of piecing apart all of the different functions that these guys are going to be able to do and what their importances are. Okay, so those are those guys there. All right, let me go ahead and go back to our PowerPoint here. Okay, any questions on these different components here? No? Okay. Okay. Is everyone able to see the PowerPoint again?
Yes.
Yes.
Yeah. Okay. Cool. Okay. So look at some of those different parts. One of the things that's kind of useful and kind of interesting here is that some of the loop of Henleys are going to be of different lengths. Okay. So some of these loops, these nephron loops here, are going to be able to extend only into the renal cortex, but some will extend as deep into the renal medulla. Okay. But in either case, all these loops of Henle are going to form a little, they're going to kind of run down. Okay. They're going to form a little hairpin loop. Okay. They're basically just kind of loop back upwards and then become the distal convoluted tubule. So the whole job of the loop of Henle is to just connect the proximal convoluted tubule to the distal convoluted tubule. Okay. And so as we look at the loop of Henle, there's going to be kind of three overall segments to it. The first part that leaves the proximal tubule is called the descending limb of the loop of Henle, or sometimes just called the descending limb. Okay. Okay. Then as it loops back upwards, okay, it's going to be the ascending limb. Okay. The ascending limb is divided up into two regions. Okay. We have a thin ascending limb, and then we have a thick ascending limb. Okay. Each of these different regions is able to allow different types of reabsorption, or reabsorption of different types of substances, which you'll learn more about in physiology when you take physiology. Okay. But just know each of these is able to do its own different thing here. Okay. So that is the loop of Henle. Okay. And so this is kind of a more detailed schematic. This is what nephrons more likely look like. It's a lot going on. There's kind of all jumbled tubes just pretty much everywhere. But again, you can see the proximal convoluted tubule leaving the glomerulus. Okay. Then we have our loop of Henle. Okay. Here's our descending limb. Then we have the ascending limb. Okay. And then we have our distal convoluted tubule. And then the distal convoluted tubule will eventually lead into the collecting duct. Okay. Then the collecting duct will eventually lead into the papillary ducts and then out as the renal papilla and so on and so forth. Okay, so that's that. So now as we kind of take a look at how these guys are built, pretty much the entire walls of the nephron are going to be made up of simple epithelium, okay? And different types of simple epithelium. Okay? So it's going to be kind of coating the entire region around the glomerular capsule, as well as the renal tubules and any ducts nearby as well. Okay? So the first thing we're going to see is the Bowman's capsule. Okay? And so Bowman's capsule is going to have some regions called podocytes, okay, some cells called podocytes. And so what these podocytes will do is they're going to sit right on top of the glomerular capillaries, and we'll see the importance of that later. Okay. But this is going to be simple epithelium to allow some of these fluids to be able to move from the glomerulus into Bowman's capsule. Okay. So they're going to have these foot-like processes here that are going to wrap around all of the glomerular capillaries. So those are the podocytes. Okay. Then we have the parietal layer here, which is just the walls of the Bowman's capsule. Okay. And so that is just made up again also of simple squamous epithelium here. And then the space in between the visceral layer from the podocyte and the parietal layer from Bowman's capsule, this space in between here is called Bowman's space. So all the filtered fluid, all the filtrate that leaves the glomerulus is going to enter Bowman's space, okay? And then from there enter the proximal convoluted tubule from there. Okay, so that's this region here. Okay, so that's all the stuff we find in Bowman's capsule. Okay. As we start moving into the rest of the nephron, the first region we're going to see is the proximal convoluted tubule. Okay? And so in the proximal convoluted tubule, which we see right here, okay, our type of epithelial tissue is going to switch a little bit. Okay? Instead of being simple squamous, it's now going to become simple cuboidal epithelium. Okay? And so this epithelial cells are also going to contain some microvilli, which are going to increase the surface area on these cells. Okay. By increasing the surface area, that increases the ability for reabsorption of different substances and then secretion of other substances as well. Okay. That's going to be in the proximal convoluted tubule. Okay. In the descending limb and the thin ascending limb, again, we're going to have simple squamous epithelium. and then the thick ascending limb of the loop of Penley, we're going to have simple cuboidal or simple columnar epithelium, which is why it becomes thick instead of thin. Okay. All right, so that's those guys there. Okay. And then when we look at the renal tubules, okay, so the part of the ascending limb of the loop of Penley, when it loops upward, it makes contact with the afferent arteriole over here. Okay, and so the cells of the ascending limb that make contact with the afferent arteriole are called the macula densa cells. Okay, so these are part of the ascending limb of the loop of Penley. Okay? These are the macula densa. Now, the cells that the macula densa makes contact with the afferent arteriole cells, okay, those are called the juxtaglomerular cells. Okay? So these juxtaglomerular cells are these muscle fibers inside the afferent arterioles. Okay? And again, they make contact with the macula densa. Okay? So together, the macula densa and the juxtaglomerular cells, or the JG cells, together they form what's called the juxtaglomerular apparatus, or the JGA. Okay, and that helps to regulate the blood pressure inside the kidneys, okay, specifically inside the glomerulus. Okay, so this is one thing that you're going to be learning about too when you take physiology as well. Okay, that's the macula densa, part of the ascending limb, and then the juxtaglomerular cells, which are part of the afferent arteriole. Okay, so they help to regulate your blood pressure. Okay, when we get to the distal convoluted tubule and the collecting duct, we're going to have two different types of cells here. The first ones are called the principal cells. And so the principal cells are cells that have a receptor for antidiuretic hormone, or ADH, okay, which we see right here. Okay. And so when these receptors bind onto antidiuretic hormone, what this does is it causes these cells to produce these proteins called aquaporins. Okay. And so aquaporins are these water channels, essentially, and so they allow water to be reabsorbed from the tubules into the blood. Okay? So if you remember, one of the roles of ADH was to help with water retention and help to retain water inside of our body. This is how that happens. Okay? So what ADH does is it binds to its receptor on these principal cells. That triggers the production of these aquaporins, and then we can now have water reabsorption happening to bring that water back into the blood. And then now we're not peeing out as much water. Okay? And we'll look at that in a little bit more detail later too. Okay. The other thing that the principal cells are going to have receptors for is aldosterone. Okay? And so aldosterone is a hormone that's going to be able to regulate your salt levels, your sodium and your potassium levels and chloride levels to some degree as well. Okay. Okay. So those are the principal cells. They're responsive to both ADH and to aldosterone. Okay. We also have another type of cell inside the DCT and the collecting ducts, which are called the intercalated cells. These intercalated cells are just responsible with your blood pH homeostasis. Okay. Okay. Any questions so far?
No.
Okay. Okay. All right. So question here. A filtrate passes through the nephron. Which of the following paths? So again, I'll let you read over that and then let me know what you guys think. All right. What do we think here?
Is it A?
B?
I
Begins
think I
with
heard A and then I heard
A.
B, I think.
Yeah, A because it
What do we
begins.
think?
It's A because it begins at the Bowman's capsule.
Right. So A is correct. So the filtrate is going to move from the glomerular capsule once it gets filtered out. Okay, then it's going to move into the proximal convoluted tubule, then the loop of Penley, and then the distal convoluted tubule, and then end in the collecting duct. So that's the path that our filtrate is going to be making throughout the nephron here. Next one. So principal cells and intercalated cells are part of which of the following? Okay, so we have the thick ascending limb of the loop of Penley, the descending limb, the collecting duct in the last part of the distal convoluted tubule, the proximal convoluted tubule, or the glomerulus. Where are those cells found?
D.
I heard D.
Oh, I said C.
Anyone else? C. Okay. Anyone else?
I think C also.
Yes, C is correct. So the principal cells and the intercalated cells, they're going to be found inside the collecting duct and the distal convoluted tubule. Okay. So that's going to be the regions where water reabsorption is going to be taking place. That's going to be the last place where water reabsorption can occur because of the action of ADH. Okay. So when we're taking a look at the nephron, there's 3 overall functions. So this is kind of where everything's going to start to come together here. Okay. So there's 3 overall functions that can occur inside the nephron. Okay. The first one is glomerular filtration. Okay. So filtration is going to be a process that takes place inside the glomerulus. Okay. And so what happens here is that the large majority of the stuff inside your blood is going to get filtered out. Okay. So water and most solutes are going to get filtered out of the glomerulus here, and it's going to get dropped off into Bowman's capsule. Okay. And so once it does that, it's then going to be able to move into the renal tubules from here. Okay. So that's the filtration process. Okay, moving the stuff from the glomerulus into the renal capsule, okay, or the Bowman's capsule. Okay. The second process is tubular reabsorption. Okay. So tubular reabsorption is a process where we take the majority of the stuff that we just filtered out and we reabsorb it back into the blood. Okay, this is going to be stuff that our body needs and things that's going to be useful to the body, and things that we want to keep inside, things that we don't want to get rid of. Okay, so about 99% of the substances that we filtered out are going to end up getting reabsorbed throughout the renal tubules. So when that happens, they're going to be removed from the renal tubules and then delivered into the peritubular capillaries, and then they can continue circulating throughout the body from there. So that's reabsorption, where we're reabsorbing all of the stuff that we want to keep inside the body, all the stuff that's going to be useful for us. Okay. The third process is tubular secretion. Okay. So secretion is the process where we remove things from the blood, we're secreting things from the blood, okay, and then delivering it into the renal tubules. Okay. So these are things that we want to get rid of, things we do not want. So this can be any waste, any drugs, or any excess like ions, any excess kind of nutrients, anything that our body does not need, okay, anything that the body does not want, or anything that may be even harmful for the body. Those are all going to get secreted out of the blood and into the renal tubules. Okay, so those are the 3 processes. Okay, we got filtration. Okay, that's going to be the stuff that we're going to be secreting out of the glomerular capsule and into the Bowman's capsule. Okay, or the glomerulus into the Bowman's capsule. We have reabsorption, where we reabsorb those substances from the renal tubules and into the peritubular capillaries, and then secretion, where we secrete things from the blood and into the renal tubules. Okay. So these are 3 processes that are really important to take place inside the kidney, because this is what helps to maintain our blood homeostasis and ultimately help us produce urine. Okay. So one way that you can kind of remember at least the difference between reabsorption and filtration is to remember that this is all going to be in regards to what happens to the blood. Okay. So we can reabsorb things into the blood. Okay. Things are going to get reabsorbed back into the blood, and then secretion is going to be where we're secreting things from the blood. Okay. So think of these 2 processes in regards to the blood itself. Okay. Okay.
So
yeah. Mm-hmm.
So everything,
Yeah, go ahead.
When it's filtered in the glomerular, all of it goes through the tubula, and that's when it's divided into whatever will be reabsorbed and whatever will be secreted. Or
yes, happens? Yeah, exactly. So, and we'll talk about the details to that in a second too as well. So just kind of a general overview of what's going to be happening. So basically what's going to be happening, like you said, is these are going to get secreted out of the glomerulus, or filtered out of the glomerulus. And then from here, the kidneys will then decide what needs to stay in, okay, what needs to stay inside the body, and then reabsorb those substances, and then what needs to be gotten rid of, okay, what needs to be excreted out, and that's going to be the secretion process here. Okay. So again, if we kind of revisit the diagram I had just drawn here. So kind of revisit this guy here. There we go. Okay. So again, here we're looking at our peritubular capillaries. Let me right hand up here. Peritubular capillaries. Okay. The first process we're going to have is filtration. Let me draw this in. I'll do this in right here. So filtration into the glomerular capillaries, or from the glomerular capillaries into Bowman space. Oh, you guys can see that. So this is, I'll just put it up here, filtration. Sorry, it's a little messy. The second process is going to be reabsorption. That's going to be where we're taking things from the tubules and delivering it back into the blood. And then we can also have secretion, where we remove things from the blood. Okay. Things that we do not want are going to get secreted out. Oh.
Okay.
Okay. Any questions so far on what's going on?
No.
Okay, just kind of quick over. Let me kind of keep this over here so you can see. Any questions before we go forward here? No? Okay. So let me move this out the way again. Okay. So those are the major processes happening inside the kidneys. Okay. So that's that. Okay. Filtration, reabsorption, secretion. Okay. All right. Everyone able to see the PowerPoint again?
Yeah.
Mm-hmm.
Okay. Okay. Oops. All righty. So here. Okay. So these are the three processes. So any fluid that comes out of this process here, that exits out of the renal tubules after all these different processes have occurred, will eventually become the urine. Okay? And so in order to determine what is going to become urine, basically what you take is everything you filtered out, you add it to whatever you secreted, okay? And then you subtract whatever was reabsorbed. Okay? Whatever is left over is going to get excreted out as your urine. Okay? So that's going to be what is considered your rate of urinary excretion. Okay? And again, the whole role of this, the whole function here, is to help regulate and maintain your blood homeostasis, okay? Namely your blood volume as well as the blood composition. Okay. Okay. All right. Now we can also have these different substances called diuretics. Okay? And so what diuretics do is they will slow your water reabsorption. Okay, so they prevent water reabsorption. The way they do that is typically by inhibiting antidiuretic hormone. Okay? They inhibit ADH. Okay? And so diuretics are usually used to treat hypertension. So people who have hypertension have obviously high blood pressure. Okay? So one way that you can kind of treat that is to eliminate the amount of water or reduce the amount of water that's getting delivered back into the blood. Okay? And so by inhibiting ADH, what you do is you basically force the water to run through the renal tubules and you don't allow it to get reabsorbed. Okay? So in doing so, two different things happen. Number one, you're reducing your blood volume because again, you're not delivering as much water into the blood. And so because you have a reduced blood volume, you're going to end up with less, fluid pressing on the arteries, and that's going to reduce the blood pressure there, okay, just by reducing the blood volume. But at the same time, it's also going to be increasing the urine production too, or your urine volume, because your water has nowhere to go. Okay, it's just going to run straight through your renal tubules and eventually become your urine. Okay. So people who are prescribed diuretics for hypertension, it does work to reduce their blood pressure, but they have to pee, quite a bit because their water is just basically getting funneled directly to the bladder, okay, through the renal tubules. So there are some naturally occurring diuretics. Caffeine is one of them. So if you ever drink like soda, for example, or coffee, these are things that will usually cause you to have to go to the bathroom pretty frequently. And then alcohol is another diuretic too. So what alcohol does and what caffeine does and what most diuretics will do is, like I said, is they're going to inhibit antidiuretic hormone. Okay. Normally what ADH wants to do is it's going to help you with water reabsorption and direct water back into the blood. Okay. However, if you're inhibiting ADH, okay, you're not going to be able to bring that water back into the blood, and it's basically just going to run straight through your renal tubules and then eventually into the bladder. Okay. And so this is one of the reasons why if you ever, are going out to the club and you're drinking lots of alcohol, okay, eventually you're going to have to start kind of going pee quite a bit, because every time you take a drink, okay, every time you have an alcoholic beverage, that reduces reduces the amount of ADH that you're producing. Okay? And if you reduce more ADH, that's more water getting funneled directly to your bladder. Okay? And so if you do that throughout the night, you're going to inhibit your ADH production, and you're going to reduce your ADH level so much that basically anything you drink is going to go straight to the bladder, and you're going to end up having to go pee constantly, even after you stop drinking for a little while. Okay? And so eventually it takes a little bit of time because it takes some time for the ADH levels to drop enough. And so that is where we get the process called breaking the seal, okay, where you have an increased urine volume because you've inhibited ADH so much. Okay? So that's how this whole process works here. I think there was something else I wanted to mention, but I think that was it. Okay. So that's how diuretics will typically work. Okay.
George.
Any questions here? Yeah, go ahead.
Yeah. Does that help people become dehydrated as well? Like, kind of drinking alcohol, and so it's
Yes.
just going to the bladder. It's not really going to their bloodstream. So it's like they're not really even getting any water in their system. So just peeing it out constantly, right? Not getting anything else. Exactly.
So Elijah's question was: is this part of the reason why people get dehydrated when they drink when they go out and drink lots of alcohol? Yeah, that's exactly right, because they're not able to retain any of the water that they're drinking. All of it just gets, like I said, it doesn't get reabsorbed. It just gets funneled directly to the bladder, and you just end up peeing it all out. So it does help to some degree to drinking water helps to kind of like filter out some of that alcohol. Okay, but a lot of times you're still going to end up feeling dehydrated either the next morning or whatever it may be, because again, you just peed out all of the water that you drank, and you didn't retain any of it because you limited your ADH production, okay, from the alcohol. Very good question there.
I have a question also.
Yeah.
For people who have high blood pressure and are on antidiuretics, are they at risk for not getting enough like calcium or all those important ions? Would that be a consequence?
Not really. Not really. Because what the diuretics do is they specifically just stop ADH. So essentially what happens is all the other ion levels stay normal, okay? And in fact, they're actually probably even, okay, I see what you're saying. So to some degree, yes, a little bit. Because water tends to want to follow like salt and things like that. And so if you're peeing out lots of water, there's typically going to be lots of like sodium and chloride and things that are also going to be getting excreted out as well. Just because you're not retaining any of that water. But usually for people who are hypertensive, you want that to happen too. Because what the sodium does is if you retain that sodium, okay, inside your body, water tends to want to follow it. And if you end up with lots of sodium and lots of water inside your blood, that's going to cause you to increase your blood pressure. So you don't want that to happen. So you actually do want to get rid of water and get rid of a few of those extra solutes as well.
Okay.
Yeah, so it is kind of beneficial to do that. Any other questions here? No? Okay. All right, so those are diuretics. Okay. So we're going to take a look at these three different processes here. And the first one is filtration. And so filtration, like we talked about, typically only happens inside the glomerulus. Okay. This is the only place where filtration is going to be occurring. And so during filtration, we're going to be forcing fluids out of the glomerulus into Bowman space. Okay. And so all the fluid that exits out is what we refer to as glomerular filtrate. Okay. So every day we filter about 150 to 180 liters of fluid. Okay. But as we know, we're not peeing out nearly that much. We're peeing out only maybe 1 to 2 liters of urine a day. Okay. The reason that we're only excreting such a small amount of urine compared to the amount that we're filtering is because we reabsorb so much of it. Okay. And we bring all that stuff back into the body. Okay. And so the way filtration typically works is, and the way it was explained to me, it kind of made a lot of sense, was every time the heart beats, it causes a lot of blood pressure. Okay? And so the glomerulus almost acts like a sponge. So every time the heart beats, the glomerulus it's almost like squeezing a sponge. Okay? And as you squeeze, you're going to squeeze all that water or out and all those solutes out of the glomerulus and into Bowman's space. Okay? And so that's where all of that filtrate is going to be ending up into. Okay? And again, having that high blood pressure, again, helps to force as much of that fluid out as possible, specifically inside the glomerulus here. Okay? And so in order to get this stuff out of the glomerulus, okay, we have to move things through a filtration membrane. Okay? So a filtration membrane is a kind of a leaky barrier that's going to be able to allow certain solids to be able to pass out of the glomerulus, but also retain other ones and prevent them from leaking out too. Okay? Specifically, the ones that we want to keep from leaking out are going to be plasma proteins and blood cells and platelets and things like that. Okay? So the filtration membrane is going to be made up of 3 different structures. The first one is going to be some fenestrations, which are these large holes and pores inside the glomerular capillary walls. Okay? And so these pores are about 0.1 micrometers in diameter. Okay, so that's large enough to allow a lot of solutes to be able to pass through. Okay, but it's still small enough that it prevents lots of blood cells and platelets from being able to get through as well. Okay, so most blood cells and platelets are about 6 to 8 micrometers in diameter, so they're way too big to be able to pass through. And so this is one way that we're able to prevent blood cells from leaking out of our kidneys and into our urine. Okay. So that's the first one. And then we're also going to have some mesangial cells inside the afferent and inferior arterioles that are able to contract. And as they contract, they help to increase the blood pressure inside the glomerulus and help to regulate filtration. Okay. So first ones are these fenestrations, these holes inside of our glomerular capillary walls. The second structure is the basal lamina. And so the basal lamina is just a layer of some tissue here between the endothelium of the glomerulus and the podocytes. And so the basal lamina is mainly negatively charged. Okay? And so again, this seems like a not really important detail, but that actually is very, very important because most plasma proteins are also negatively charged. Okay? And so what this does is if you have plasma proteins that are negatively charged, you have a basal lamina that's negatively charged, they're going to repel each other, and that's going to prevent the plasma proteins from wanting to get anywhere near the walls and end up accidentally leaking out. Okay? Because they're negatively charged, they're going to repel each other, and so the plasma proteins are going to stay retained inside the glomerular capillaries. Okay? That's number 2. And then the third structure are these pedicels, which are the end process, the foot-like processes of the podocytes. Okay? And so here would be one pedicel, here's another pedicel. So all these little legs here are these pedicels. And so the pedicels are also called filtration slits, will kind of form what's called a slit membrane. Okay? That slit membrane is very, very small, very, very narrow, about 0.007 micrometers in diameter. Okay, so again, that is a very small diameter that allows very small molecules to be able to pass through, like water, various ions, in some cases, very small plasma proteins, in very rare cases, ammonia and things like that. Okay. That's going to allow very small molecules to be able to pass out of there, but prevent other larger molecules from being able to exit through the slit membranes too. Okay. So those are the ways we're going to be able to prevent some molecules from being able to get out while allowing others to get out as well. Okay. So if the glomerulus becomes inflamed, that can lead to a condition called glomerulonephritis. Okay? And so what happens in glomerulonephritis is usually caused by an allergic reaction to a streptococcal bacteria, the one that causes strep throat, or a family of the ones that cause strep throat. And so what happens here is these glomeruli become really inflamed, and they become really swollen, and they become so full of blood, and the pressure becomes so high that they start to push blood cells and plasma proteins out of the glomerulus and into your urine when they shouldn't be there. Okay. And so you end up with conditions like hematuria, which is blood in the urine, or proteinuria, where you have protein in the urine, and some red blood cell casts as well, which you'll see here. Okay. So this is what happens if the glomerulus becomes inflamed. Okay. Blood and proteins should not be entering the urine, but if you have some kind of kidney failure or the glomerulus becomes inflamed, this is some of the things that can take place. Okay. Okay. Okay, so that's filtration. Okay, any questions on filtration before we go forward?
No, not really.
Okay.
Uh,
Okay. So then the next. Oh yeah, go ahead. Third question.
so the substances that are left in this membranes, are they just gonna stay there?
Pretty much. So unless this gets treated, then yeah, it would pretty much just stay just like that, and things would continue leaking out when they shouldn't be. Yeah. Okay. Okay. So that was filtration. Okay. The next process that happens after filtration is going to be reabsorption, and so this is the process where we start to reclaim some of those materials that we filtered out and bring them back into the blood. Okay. Bring them back into the body. So it's reabsorption because you're almost like you're reabsorbing it back into the blood. Okay. So reabsorption is a process that takes place where we start to reabsorb all the things that we want to continue to have inside of our blood, things that are useful, the things that we need inside our body. And so reabsorption is going to happen throughout the renal tubule. It's going to happen in the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and in the collecting duct. However, the proximal convoluted tubule is going to be the place that has the most reabsorption taking place. Okay? The proximal convoluted tubule has the largest contribution to reabsorption. Okay? Okay. Everything else does, reabsorption, like I said, does happen everywhere else, but it's going to happen mostly inside the proximal convoluted tubule. Okay. So some of the substances that get reabsorbed are going to be glucose, sodium, potassium, calcium, chloride, bicarbonate ions and phosphate ions, and then to some degree, some small proteins and some small peptides can be reabsorbed as well. Okay? And this is going to be done through various types of passive and active processes. So again, these are things that we want to retain inside the body. Okay? However, there is a condition that can take place here. And so if, for example, we have too much blood glucose inside of our body, let's say our blood glucose levels are about 200 milligrams per milliliter. Okay? That's too high. It should be only about 90 or so, 85 to 90 or so. Okay? But if our blood glucose levels get to about 200 or more, then what happens here is the proximal convoluted tubule can't reabsorb the glucose fast enough. Okay? And so if that glucose doesn't get reabsorbed fast enough from the proximal convoluted tubule, because that's the only place where glucose can be reabsorbed, if it doesn't get reabsorbed fast enough, that glucose just continues to stay inside the filtrate and eventually becomes part of the urine. And so that creates a condition called glucosuria, which translates to glucose in the urine, okay, which should not be taking place. We should be able to absorb or reabsorb most of that glucose inside the proximal convoluted tubule. Okay, but again, if our blood glucose levels get too high, we can't reabsorb it fast enough, and we end up getting glucosuria. And this is a condition that leads to that's caused by diabetes. Okay, and so in diabetes, one of the things that people will see is excess glucose production inside the urine. So is it, actually, I don't know if I brought this up last week, but does anyone know how, like way back in the day, people used to test, or doctors would test and diagnose diabetes? Anyone know how diabetes used to be diagnosed, a long time ago?
No.
Anyone know?
Not really. Does it have to do with something like
No. So what doctors used to do in order to diagnose diabetes is they would actually taste their patient's urine. Okay? And if the urine tasted sweet, then they knew there was excess blood glucose, or there was a high level of glucose in their blood, which caused them to have a high level of glucose in the urine. So luckily we've come a long distance from that. But that was essentially how doctors, like way back in the day, used to diagnose diabetes. And so some of the conditions that can take place with this happening is because you have excess glucose in the urine that causes an inhibition of water reabsorption. And so because you have lessened water reabsorption, you're just producing more and more urine. And so you end up with what's called polyuria, or like excessive urine production, or excess urine volume. And so that's one of the first signs of someone dealing with diabetes, is that they'll start to pee constantly, and in some cases they'll start wetting the bed pretty frequently, because they're just producing so much urine because of this excessive glucose production in their body. And so they end up with decreased blood volume because, again, they're not reabsorbing enough water, and then dehydration too. So that is reabsorption. Okay. Any questions on reabsorption?
No.
No? Yeah. So, yeah, go ahead.
Does this have anything to do with gestational diabetes?
Say that one more time. With gestational diabetes, is that what you said?
Yes.
Yeah. So gestational diabetes. So her question is, does this have anything to do with gestational diabetes? And I believe it does. Because what happens, so in some cases, some mothers, even though they may not have been diabetic throughout their life, during a pregnancy, they'll start to become diabetic. And some of these processes will kind of continue to occur. So in some cases, what I don't know the exact basis behind it, but my guess is their bodies, because their bodies are changing, they're not as good at, like retaining that glucose and storing that glucose. And so it kind of stays inside their blood. And so because their blood glucose stays at a higher level, this process continues to occur and they can't reabsorb that glucose fast enough and they end up with gestational diabetes. That'd be my guess, but I'm not entirely positive on exactly how that works. But then after the pregnancy, in some cases, it usually goes away and you kind of go back to not being a diabetic. But I do believe in some kind of more rare cases that diabetes kind of continues to kind of be a factor that they have to deal with too. Yeah. Good question. Any other questions here? Okay. So that's reabsorption. Okay. And then the third process is secretion. Okay. So tubular secretion is the process where you secrete extra substances from the peritubular capillaries into the renal tubule. So this is all the stuff you want to get rid of. Okay. Anything that you either were not able to filter out before, or anything else that just needs to just get out of the body. Okay. And so some of the things that can be secreted out would be hydrogen ions, which will help to monitor your blood pH levels, some potassium, some ammonium ions, creatinine is another one, and then some different types of drugs like penicillin. They can all be excreted or secreted from the peritubular capillaries into the renal tubules, as we see here. Okay. So secretion, like we said, has two overall roles. Number one is to regulate your blood pH by secreting hydrogen ions out. Okay. And then also by secreting excess substances and getting rid of any extra waste that you don't need in your body. Okay. And then from there, all of those substances will then enter into, or the filtrate after secretion has occurred, will then enter into the collecting duct to eventually become the urine. Okay. So that is secretion. Okay. Any questions on secretion before we go forward here? Okay. Well, this is just kind of a little schematic here. We'll basically, and I want you to watch this video on your own time. But basically what happens here, again, our first function is going to be filtration. That's going to happen in the glomerular capillaries. We're going to filter their blood out into the Bowman's capsule. Okay. Certain molecules are going to get reabsorbed. About 99% of the filtrate gets reabsorbed back into the peritubular capillaries to eventually be retained inside the body. Other things will be secreted out from the peritubular capillaries and into the renal tubules. Okay. And then whatever comes out of this process becomes your urine. Okay. Okay, so question here. Which of the following statements is false? We have A, the first step in urine production is glomerular filtration. B, in tubular secretion, substances not needed by the body are moved from the blood to the tubules. C, fluid that enters the capsular space is glomerular filtrate. D, in tubular reabsorption, water and useful solutes are reclaimed into the blood. Or E, the diameter of the efferent arteriole is larger than the afferent arteriole.
E.
Which one is false?
I think E is the opposite of statement. So it should be
I think
E.
I heard D and I think I heard E. Yes, E is correct. So E is false. So the diameter of the efferent arteriole is not larger than the afferent arteriole. The efferent is smaller than the afferent, and again, that helps to create that extra blood pressure inside the glomerulus. Okay, good. Okay, so that's how we end up forming that urine. Okay. Any questions on the urine formation here?
No, not right now.
No? Okay. Okay. right. So once all those filtrate starts to leave the collecting duct, okay, it's now going to become considered urine. Okay? And so that urine is going to leave the collecting duct. It's going to enter the papillary duct, and then make its way into the minor calyces, into the major calyces, into the renal pelvis, and then make its way into the ureter. Okay. And so then from there, the ureter is then going to transport that urine into the bladder, and then it's going to exit through the urethra. Okay. So we're going to look at the ureters first. And so the ureters, like we talked about earlier, their main job is to just transport the urine from the kidneys down to the bladder. And so in order to get into the bladder, there's going to be two holes here, two holes at the posterior aspect of the bladder, which are the ureteral openings. And so these kind of open at an angle. Okay. So the ureters are going to enter into the bladder at an angle from the posterior side of the bladder, and then drop off that urine there. Okay. As the bladder begins to fill up with urine, the pressure inside the bladder is going to begin to increase, and that's going to cause these openings here, these oblique openings here, to close up, okay, and compress. And so that becomes useful because that helps to prevent the backflow of urine up through the ureters. Okay. Now, the reason this is helpful is because if there's any bacteria that happen to make their way inside the urine, okay, if these openings don't close properly, that can cause the bacteria to be able to enter back up into the ureters and then make their way back up into the kidneys, and then all of a sudden now you got kidney infections. Okay, so we need those openings to be able to close up properly, as the bladder begins to fill with urine. Okay. This is what this whole process, what these look like here.
Those
So
are
here's the ureter openings. Yeah, go ahead.
they're the oblique openings.
Yes. Yeah, so these are the ureteral openings here. So we have 1, 2 here. And so these will compress as it gets, as the bladder begins to fill, and prevent any urine from flowing backwards into ureters.
Oh, okay.
When we look at the ureters, there's going to be a mucosa region. And so that mucosa is going to be made up of mostly transitional epithelium. And so if you remember going back to chapter 3, transitional epithelium is able to switch back and forth from squamous cells to cuboidal cells. And that allows that particular organ to be able to stretch, okay? To stretch to accommodate some kind of substance that's entering it. Okay? In this case, inside the ureter, we're going to have that same type of epithelium. We're going to have that transitional epithelium, which allows the ureters to be able to stretch to accommodate the urine flowing out of the kidneys. Okay. And then it's also going to be producing some mucus, and the mucus just helps to prevent the ureters from coming into direct contact with the urine, and so because the urine can become pretty acidic. Okay? So if there's extra acid there, and we didn't have that mucus, it could cause some significant damage and irritation to the ureter's walls. Okay? So the mucus just helps to protect the ureter's walls from being exposed to too much acid inside the urine. Okay. So the ureters will then lead into the bladder. So the bladder is going to be a pretty hollow, muscular organ that's able to stretch. And it's going to be found inside the pelvic cavity. And it's going to be found posterior to the pubic symphysis. Okay. So in males, the bladder is going to be anterior to the rectum. In females, it's going to be slightly smaller, but it's going to be anterior to the vagina, and it's going to sit up underneath the uterus. Okay. It's going to be inferior to the uterus. So typically the bladder is able to hold about 700 to 800 milliliters of urine. And as it swells, okay, as it starts to fill, it's going to be able to change shape, okay, and be able to swell up and in order to accommodate that urine. Okay. And that's going to be due to transitional epithelium inside the walls of the bladder. Okay. The bladder also has a triangular region called the trigone. And so the trigone is made up, it's kind of formed by the two ureteral openings, okay, 1 and 2, as well as the internal urethral orifice, okay, which is the opening that leads into the urethra. Okay, so these 3 points here form the trigone. Okay, 3 openings inside the bladder. Okay. Okay, that's that. Okay, so this is what it would look like inside of a female, in a woman's cadaver. here we have the bladder, and then again, sitting right on top of the bladder, you would have the uterus. Okay. And then anterior to the bladder, you're going to find the vagina here, okay, and so on and so forth. So we'll take a look at those later on. Or sorry, yeah, got that right, cool. Okay, so that's the bladder. Okay, so when we look at the bladder, there's gonna be 3 overall coats to it. The first one is called the mucosa. So inside the mucosa, that's gonna be the deepest layer, and that's gonna form the walls. We're gonna find our transitional epithelium, we're gonna find some lamina propria, and then we're gonna find some rugae, which is the ridges that are kind of similar to the ridges we saw inside the stomach. Okay. So we're gonna have some rugae here inside the mucosa. Okay. Then we have the muscularis, which is also called the detrusor muscle. Okay. And so this muscle is able to contract in order to allow us to pee, and we'll look at that later. Okay. Inside the detrusor muscle, we're going to have the internal urethral sphincter, which is involuntarily controlled. Okay. And then we have the external urethral sphincter, which is made up of skeletal muscle, and this is one that we are able to control. Okay. And then on the outside of the bladder, we're going to have the serosa. Okay. So we have the mucosa, we have the muscularis, which contains the detrusor muscle, the internal and external urethral sphincters, which we see over here, and then we have the serosa on the outside. Okay, cool. All right, cool. So that's just a picture of the histology of what the bladder would look like. Again, you have serosa, you have the muscularis down here, and then the mucosa, and this will be the lumen of the bladder up here. Okay. All right, any questions on the structure of the bladder before we go forward? No? Okay. Okay. So, as we know, the bladder is going to be an organ that's going to help us with urination. Okay. This is going to lead us to the micturition reflex, or also called urination or voiding of the bladder. And so micturition is just the removal of urine, or the excretion of urine from the bladder here. Okay. So in order to cause micturition to take place, the first thing that's going to happen is the bladder obviously has to fill with some urine. Okay. So once that bladder fills with urine to about 200 to 400 milliliters of urine, the pressure inside the bladder is going to increase dramatically. Okay. And so as it increases, that's going to cause some stretching inside the bladder, and that's going to cause the stretch receptors inside the bladder's walls to fire and become activated. Okay. So these stretch receptors are going to send action potentials down to the micturition center inside the spinal cord. Okay. And that's going to trigger the micturition reflex. Okay. Okay. So again, all we had here was just some stretching happen. Stretching causes these stretch receptors to activate. Okay. And then those are going to send action potentials down to the micturition center inside the spinal cord. Okay. From there, we're going to have some parasympathetic impulses here. Okay. And so these parasympathetic impulses coming from the micturition center are going to send motor impulses down to the bladder, okay. Specifically the detrusor muscles of the bladder, and as well as to the internal urethral sphincter. Okay. And so what's going to happen here is these motor impulses are going to cause the detrusor muscle to contract and cause the bladder to contract. And at the same time, it's going to cause the internal urethral sphincter to relax. Okay? And so that's going to allow urine to be able to begin to exit out of the bladder here. Okay? Now, when we are ready to pee, okay, what's going to happen is we're going to have an inhibition of somatic motor neurons that stimulate the external urethral sphincter. Okay? And so when we inhibit those motor neurons, that's going to cause the external urethral sphincter to open as well. Okay? And so that is going to allow us to be able to pee, okay, and urinate. And so just kind of recap here. Okay, we have our bladder beginning to fill. Okay. As the bladder fills, we have our stretch receptors getting activated. They're going to send action potentials down to the spinal cord. And we're going to have parasympathetic impulses, okay, sending those action potentials down to the motor, to the detrusor muscle, causing the muscle of the bladder to contract, and also causing the internal urethral sphincter to relax, which you see over here. Okay. And then when we are ready to pee, we can voluntarily relax our external urethral sphincter, okay? And that will cause kind of another opening, and that's going to allow pee to flow out of the urethra and then into the toilet, ideally. Okay. So in order for urination to take place, we need contraction of the bladder, and then we need relaxation of both sphincters. Okay. But again, we need parasympathetic impulses, even though, again, sometimes it may seem like it's a sympathetic situation here. Okay. So these are going to be parasympathetic impulses regulating this whole process here. Okay. Any questions on the micturition reflex?
No.
Fairly similar to the defecation reflex. Fairly similar kind of pathways, similar patterns, basically working almost the exact same way. Okay. Any questions here? No. Okay. All right. So as the bladder begins to fill with urine, it starts to create a sense that, that the bladder is beginning to fill. And that sensation starts to appear right before or well before the micturition reflex actually takes place. And so this whole process begins to tell your brain that, hey, it's time for us to go to the bathroom because, you know, if we don't go pretty soon, we're going to be wetting our pants here pretty soon, and we don't want that to happen. Okay. So it creates a conscious desire to go to the bathroom before that micturition reflex actually kicks in. Okay. And then again, what's going to happen here is the external urethral sphincter and then specific muscles around the pelvic floor can be voluntarily controlled to either stop you from peeing or help you pee, you know, when you're ready. Okay, so that's the bladder. Okay. Now, when people are not able to control the bladder, that is what's called incontinence. Okay, so incontinence is the lack of control over micturition. And so this can happen for a number of reasons. The two most common reasons are what's called stress incontinence and then urge incontinence. Stress incontinence typically happens when you're dealing with either young or middle-aged women who just have less control over their bladders just because they've had a baby. And so because they had a baby, some of the muscles around their abdomen and around their pelvic floor are not as strong. And so sometimes it can cause them to kind of, you know, pee a little bit, whether they sneeze or whether they laugh a little bit too much. That pressure that happens causes them to release a little bit of urine and cause them to pee a bit. And then we can have urge incontinence, which typically happens in elderly people. And this is just when they have a very sudden, kind of urgent or abrupt, intense urge to go pee. And then all of a sudden they just lose the ability to hold it, and then they end up peeing. So those are the two major types of incontinence, although there are a number of other causes of incontinence as well. Okay. Then the last one we have is the urethra. And so the urethra is the little tube that's going to help deliver the urine from the bladder into the toilet. Okay. And so the urethra is going to run from the internal urethral opening all the way up to the exterior of the body. And so in females, the urethra is typically relatively short compared to the male's urethra, and we'll look at that in a second. And it's going to end at the external urethral orifice here, which sits in between the clitoris as well, the clitoris and the vaginal opening. Okay. So that's going to be where the external urethral orifice is going to be found for females. Okay. And that's going to be the location where the pee is going to be exiting out of the body from. Okay. In males, this process is a lot more complicated. Okay. The male urethra is a lot longer, and it gets divided up into three different regions. The first one is the prostatic urethra, and so that is going to be where the urethra passes through the prostate. Okay. Then from there, the urethra will then pass into the the perineal muscles, and so that is going to be what we refer to as the intermediate urethra or the membranous urethra. Okay. And then, that's what we see down here. And then we have the spongy urethra. And so the spongy urethra passes through the penis. So that will ultimately lead out to the external urethral orifice, which is at the tip of the penis. And then again, that's going to be where the pee will exit out of the body from. Okay. Now, the other thing about the urethra is that this is also going to be the same passageway that semen is going to be using to exit the body from as well. And we'll look at that on Wednesday when we look at the reproductive system. Okay. So as you can see, the male urethra is a lot longer and a lot more complicated than the female urethra is. Okay. So this is a picture that kind of just shows the comparison between the two side by side. And I believe That is it. So that's just a picture of the bladder. Last thing here. So urinalysis. So one of the things that's really important is to be able to analyze your urine, because analyzing the urine tells you a lot about what's going on with someone's health. And so you can look at the volume of the urine, you can look at the physical, chemical, and microscopic components of the urine. And depending on what you find, that can tell you different aspects or different characteristics about that person's health. There's different types of urinalyses. And if you take physiology, hopefully next semester when we're all kind of back on campus, you'll be able to do this in Bio 9 as well. Okay. And so you can either do like a complete urinalysis where you take a sample of the urine and you look at it under a microscope and look for specific characteristics. You can do a rapid urinalysis where you take a little like a dipstick that has a bunch of pads that will change color depending on different concentrations that exist inside the urine. And then you can do a 24-hour urine collection where you have someone pee in a cup basically over a 24-hour period. And that gives you a more complete view of what's going on with their urine, what's going on with their kidney health and things like that. Okay. So this helps to determine various different diseases that someone may be dealing with, or various different types of renal condition that someone may be having, or a number of other types of conditions too. So urinalyses are very, very important. And so if you were ever to go see a urologist, this is the first thing that they'll have you do right off the bat before you even go and see the doctor. That'll be going pee in a cup, and then they'll analyze the urine before you even go and see the doctor from there, or the urologist. Okay. Any questions so far? Any questions on anything we've talked about so far?
No.
No.
Okay. So last question here. So which of the following is true of the micturition reflex? Okay. So we have A, parasympathetic impulses cause contraction of the internal urethral sphincter and relaxation of the external urethral sphincter. B, parasympathetic impulses cause contraction of the detrusor muscle and relaxation of the internal urethral sphincter. C, parasympathetic impulses cause relaxation of the detrusor muscle and relaxation of the external urethral sphincter. D, sympathetic impulses cause contraction of the detrusor muscle and relaxation of the internal urethral sphincter. And then E, sympathetic impulses cause contraction of the detrusor muscle and contraction of the internal urethral sphincter. And which one is true?
B.
I hear B. Do we agree? Do we disagree? Anybody?
I'm not
Well, B is correct. Yeah, so B is correct. So what's going to happen here is the detrusor muscle is going to contract from the parasympathetic impulses, and then it's also going to cause the internal urethral sphincter to relax as well. All right, good. All right, that's it for everything we've talked about today. All right, that went a little longer than I was expecting it to go. But are there any questions on what's going on here?
I was gonna ask you, I did see that you uploaded some, are those our old exams? I didn't have a chance before class to open them. Is that
Okay.
our old exams?
Yes. Yes. So those are copies of the exams we took earlier this semester. So you can use them to study for the exam. So here you have the exam 1 key and the exam 1, exam 2 and the exam 2 key, exam 3 and the exam 3 key. I will put up exam 4 this week, or I'll do that tonight, so you guys have that as well to study for, and then that way you can kind of have an idea of what to study and something to kind of have background information on.
Okay.
However, what I did decide to do is on the day of, actually maybe not. Let me think. Let me think before I start saying stuff that may not actually be true. Ignore what I just said. But yeah, this is all stuff. This is all here for you, and I'll upload exam 4 as well.
Okay, because I was thinking, since we're gonna have the review the Monday before our last exam on a Wednesday, it will be helpful if we start moving on these, like reviewing them and filling in our answers, right? So we know, or how will
Yes. Yes, so it'd definitely be useful to look over those. Because like I said last week, for the final, the first 50 questions are going to be on new stuff. The next 40 questions from 51 to number 90, those are all going to be from the previous exams. And so I'm going to be pulling questions from previous exams, from anywhere on the exam. So I might have taken a free response question on exam 4, for example, and made it into a multiple choice question for the final. Okay. So go through those different exams so that you can, really be up on it and really make sure that you fully understand everything. So that way you can, make sure you do well on the final.